Hello everyone. Today we are going to discuss the development of the brain, and this is a further lecture after we've covered the development of the spinal cord. The link to the YouTube video will be pinned in the in the comment section. So at this point, let's discuss the development of the brain, and the scope for the session will be trying to recap what is neurulation. We'll recap neurulation in terms of primary neurulation and secondary neurulation. We'll also try to describe the primary and secondary brain vesicles, and then we'll also describe the derivatives of those brain vesicles. And then we'll also describe the common malformations of the brain. These are the congenital anomalies of the brain that come due to these brain vesicles. And then we'll try to highlight the factors which influence the brain development. And then finalize with some of the sample essay questions that we might be asked in an exam setting. So, what is neurulation? Neurulation at this point is a formation and development of the neurotube from the ectoderm. So, of course, when you talk about ectoderm, you're talking about the three germ layers: the ectoderm, mesoderm, and the endoderm. So, ectoderm gives us the neuroectoderm, then the neuroectoderm forms the neurotube. So, the origin of this neurotube is that it comes from ectoderm, specifically the neuroectoderm, and then also the secondary neurotube comes from secondary neurulation, comes from mesoderm. The undifferentiated cells of the mesoderm, that is the mesenchymal cells that it contains, to give us the neurotube. So just to remind us of the three types of neurulation, you have primary neurulation that comes from the ectoderm, the neuroectoderm. Secondary neurulation that comes from mesoderm, that is the mesenchymal condensation. Then you'll have junctional neurulation that comes from joining of the primary and secondary neurulation. So we'll try to describe primary neurulation. Just a recap of what we covered some times back is that you have the three germ layers. You have the ectoderm, you have the mesoderm, and then you have the endoderm. In the mesoderm, you have this notochord. The notochord induces the ectoderm to become the neuroectoderm. So the thickening will happen at this point to give us a neuroplate. Then the neuroplate will undergo the process of folding to give us the neurogroove. Is a well-demarcated neurogroove. Then fusion will happen. And these folds to give us a neurotube, and then the cells of the folds will disintegrate to give us the neuronuclear cells. For secondary neurulation, we have the three germ layers. We have the ectoderm, mesoderm, and then the endoderm. What will happen is that this mesoderm, which is the mesenchymal cells, the undifferentiated cells of the mesoderm, will condense, will aggregate. This is the mesenchymal condensation that I talk about. Give us a neural rod, or the medullary rod is a neural rod. So this neural rod will undergo mesenchymal epithelial transition to give us a cavity. So this cavitation that we talk about. So remember, the size of the mesenchyme can change to epithelium, or they can change from epithelium to mesenchyme. So that is epithelial mesenchymal transition and vice versa. So what is happening at this point is that these cells of the mesenchyme will change to epithelium. So that is mesenchymal epithelial transition to give us a cavity, the central canal of the secondary neurotube. So when you form a cavity, you've already formed your neurotube, which is definitive at this point. Yeah. So what are some of the differences between primary neurulation and secondary neurulation? The first difference is that primary neurulation will have the notochord inducing this process of neurulation, but on this other end, we won't have the notochord inducing process of secondary neurulation. Then the other one is that primary neurulation will be coming from the ectoderm, secondary neurulation will be coming from the mesoderm. Primary neurulation will give us the neural cells. Most of the neural comes from primary neurulation. Secondary neurulation give us none or very little neural cells. The other difference is that primary neurulation gives us the proximal part of the neural tube, and then secondary neurulation gives us the caudal part of the neural tube. That is to mean then that the primary neurulation will form the brain and some part of the proximal spinal cord. Then secondary neurulation will form the caudal part of the spinal cord. So those are about 5 differences of primary and secondary neurulation. Maybe another difference that you can add up is that we'll have the epithelial mesenchymal transition happening, the mesenchymal epithelial transition happening in secondary neurulation while it doesn't happen in primary neurulation. Yeah, then presence of notochord, absence of notochord on primary neurulation, then it's absent in secondary neurulation. Yeah. So with this now we can look at this ectoderm here. We have outer ectoderm with the neuroplexus and we have the neurotube. So the ectoderm, we discussed this when you're talking about the development of the integumentary skin. So it will give us the epidermis, hair, nails, sebaceous glands, the olfactory epithelium, the mouth epithelium, and the lens, the cornea. But the neural cells will give us the peripheral nervous system, the Schwann cells, the neuroglial cells, and the ventricular cells will give us the sympathetic nervous system and the parasympathetic nervous system. It will also give us the chromaffin cells of the adrenal medulla, give us the melanocytes and the Merkel cells. It will give us the facial cartilages. It will also give us the dentine of the teeth and even the odontoblast cells of the teeth. The neurotube, it will give us, we're going to discuss today, it will give us the brain, the neural part of the pituitary gland, that is in the neural hypophysis, will give us a spinal cord, the motor neurons, and the retina. So these are neurotube that I'm discussing today. So remember, the primary neurotube will give us the brain and proximal part of the spinal cord, while the caudal neurotube will give us the caudal part of the spinal cord. So the proximal parts of the neurotube will undergo enlargement. It will develop faster than the spinal cord to give us 3 brain vesicles. The 3 brain vesicles will be the prosencephalon, mesencephalon, and rhombencephalon. The prosencephalon representing the forebrain, mesencephalon representing the midbrain, and then rhombencephalon representing the hindbrain, this point. So we realize that we have canals in them. So this canal will form structures, the ventricles and the various canals in the brain that we'll discuss in a few minutes. So prosencephalon forms the forebrain. Mesencephalon is actually the midbrain, then the rhombencephalon. They will give us the hindbrain as we'll see in a few minutes. But before then, if you can cut the neurotube transversely, this is what you can appreciate. It will have 3 layers, the germinal layer, or the ventricular layer, it will have the mantle layer, or the intermediate layer, then it will also have this marginal layer, the outer layer. So you can appreciate the notochord and the neural plexus at this point. So what it will differentiate into this, it will differentiate by first of all giving us the basal plate, which has motor functions, it will have motor nuclei, and then this other plate will have sensory nuclei, it will have sensory functions. And then this central canal is one that will now differentiate to give us the various ventricles, such as the 2 lateral ventricles, the 3rd ventricle, the cerebral aqueduct, and the 4th ventricle, and even the canal of the central of the spinal cord will be coming from this central canal that is present. So we now narrow down, we've talked about the 3 primary brain vesicles. The 3 primary brain vesicles are actually a prosencephalon, mesencephalon, and rhombencephalon. So at around week 3 to week 4, so the 3 to week 4 embryo will have 3 brain vesicles. So at around now week 5, the prosencephalon, the mesencephalon, and the rhombencephalon will differentiate. So we'll appreciate that prosencephalon will differentiate telencephalon and diencephalon. Mesencephalon will continue as a mesencephalon. Rhombencephalon will divide into two to give us metencephalon and myelencephalon. So telencephalon will give us the cerebral hemispheres. So the cerebrum will be coming from telencephalon. Then the diencephalon will give us the thalamus, the hypothalamus, epithalamus, subthalamus, and even the metathalamus will be coming from the diencephalon. So 5 structures: thalamus, hypothalamus, epithalamus, subthalamus, and metathalamus will be coming from the diencephalon bit. Actually, those are the components of the diencephalon. Then mesencephalon will continue to give us the midbrain, and then the rhombencephalon, which divides into which differentiate with metencephalon and myencephalon, in find that the metencephalon will give us the pons anteriorly and then posteriorly to give us the cerebellum, and then the myencephalon will give us the medulla oblongata. So the neuronal that is present in this is that it will differentiate. So for example, the canal, the neural canal that will be present in the cerebrum will be the two lateral ventricles. Then the diencephalon will have the third ventricle, and then in between, in the middle, you'll have a cerebral aqueduct. In the metencephalon, we'll have, so in between the metencephalon and then also the pons and the cerebellum will have a fourth ventricle there, which extends up to the level of open medulla, that is the fourth ventricle. Then after that, knowing the level of closed medulla, you'll have your central canal going downwards in the spinal cord. So with that, actually, we can have the ventricular system in this. That ventricle, the lateral ventricles in the cerebrum will communicate with the fourth, with the third ventricles that is present in the encephalon, using the interventricular foramen of Monro. Then you'll have the fourth ventricle communicating with the third ventricle using the cerebral aqueducts. And then after that, we'll have now communication either going to the subarachnoid space or to the central canal of the spinal cord using the various foramina that will be present. So what is the function of the ventricular system? Just stores and allows even circulation of CSF, and even production of CSF happens in the ependymous end that lines those ventricles. So given the fact that you talked about these 3 and 5 brain vesicles, we need, and their derivatives on the right side, we need to describe the brain flexions. So by the 4th week, so this is the 4th week at this point, the neurotube grows rapidly. So it's growing rapidly. And due to that, now it will bend ventrally. The reason you can appreciate it's bending ventrally at this point, producing 2 flexures. And the 2 flexures is that you have one at this point. This is a cephalic flexure. Some books will call them the midbrain flexure, the level of the midbrain. And then the other one will be the cervical flexure. And the cervical flexure will be present now differentiating the hindbrain in the spinal cord. This is a cephalic flexure. Those one will be like primary brain flexions. Then in the secondary brain flexions, we'll have one, the potent flexure that appears in the hindbrain. And this one will be opposite to the cephalic and the cervical flexions. So what will happen is that it will lead to thinning of the roof of the hindbrain. So the roof of the fourth ventricle will be very thin, and that's the reason it will have its thinning. So the potent flexure will appear posteriorly, then the others will appear anteriorly. So the 3 brain flexions will have the cephalic flexion, the level of the midbrain, cervical flexion, differentiating the hindbrain and the spinal cord, the pons inflection now, acting posteriorly, compress the wall of the hindbrain, leading to the limb of the roof of the hindbrain. So that's the brain flexures you can appreciate in here. The cervical flexure, differentiating the spinal cord and the hindbrain. You have a pontine flexure acting posteriorly of the mesencephalon at this point, and then the cephalic flexure will be acting on the midbrain. And actually, you can appreciate this point is a mesencephalon. So we need to appreciate that here at this point you have the diencephalon with the lateral ventricles, is the oblongata terminalis. This diencephalon at this point it will develop optic cups, and then these optic cups will be going to develop to give us actually our future optic nerves and optic tracts at this point. So this diencephalon will give rise to the optic nerve at this point. Then the mesencephalon will give rise to the pons. The cerebellum encephalon will give rise to the medulla oblongata. Of course, we've talked about the derivatives of the center of the neural canal that is present in there. So a better representation of that. So this is what you can find in an exam setting. You can find the telencephalon giving us the cerebral vesicles that will bring include cerebral hemispheres, and then this diencephalon giving rise to the optic stalk at this point, the midbrain, the mesencephalon, the hindbrain, giving us the metencephalon that is on the pons anteriorly, and then the cerebellum posteriorly, and then the mesencephalon giving us the medulla oblongata. Then the remaining that will be the spinal cord. For the flexions, you can appreciate at this point, we'll have the cephalic flexure available in the midbrain. We'll also have our pons inflection acting posteriorly at this point. Okay, so those are the derivatives. We've seen the three primary brain vesicles, the forebrain, the midbrain, and the hindbrain, that is the prosencephalon, mesencephalon, and rhombencephalon, giving rise to the forebrain, prosencephalon giving us telencephalon, diencephalon, and then the midbrain give us the mesencephalon. Hindbrain will form the diencephalon and myelencephalon, and the derivatives. So those are the 5 secondary brain vesicles. What are the other derivatives? So the diencephalon will give us the cerebrum, hippocampus, and the olfactory lobes. So olfactory lobes is for smell, hippocampus is for memory storage. Cerebrum is actually for association. So talk about intelligence, all these cognitive functions happen in the cerebrum, even motor functions, occipital functions. So according to which lobe will form, their functions will be there. Then for the diencephalon at this point, give us the retina for vision. The thalamus, the pineal gland at this point, the biological clock. The thalamus acts as the relay center for optic, auditory, and even other tracts. The other neuron comes from the thalamus. The hypothalamus at this point regulates temperature, sleep, breathing, regulation, sympathetic output, and other structures. Not forget that you'll have the subthalamus and the metathalamus, so coming from the diencephalon. For the mesencephalon, you'll have the midbrain. So this one acts like a connection between the diencephalon and the remaining part of the brainstem. So fiber tracks between the anterior and posterior brain, and then even the optic lobes and the tectum will contain water structures. Not forget that it also contains substantia nigra structure that has a subset on the striatum. So we remember for the mesencephalon, it will give us the pons anteriorly and cerebellum posteriorly. So pons at this point is a fiber tract between cerebellum and cerebellum. So pons means a bridge that will connect the cerebrum and the cerebellum. the fiber tracts the pontocerebellar tract, the cortical pontocerebellar tract. For the cerebellum, just for coordination of complex muscular movements happen in the cerebellum. For the medulla, it contains vital centers such as vasomotor centers, the cardiorespiratory centers, and the thermoregulatory centers will be present in the medulla. And even the metic center will be present in the medulla also. So what are some of the anomalies of this brain? The first one is hydrocephalus. So hydro means water and cephalus means brain. So enlargement of the ventricles. So this enlargement of the ventricles may be due to obstruction of the ventricular system, the one that we described, or overproduction of cerebrospinal fluid. So to do that now, you can have two types of hydrocephalus, obstructive and non-obstructive. So obstructive is when you have stenosis or maybe complete obstruction of the ventricles or maybe other canals. Then in communicating or non-obstructive hydrocephalus, that is when you have mutation of the choroid plexus, mutation of the ependymal cells producing a lot of CSF, but the normal circulation is being maintained. So remember hydrocephaly is amboxy lymphatic hydrocephalus can have two types, communicating stroke non-obstructive or obstructive stroke non-communicating hydrocephalus. Polycencephaly is when you have the normal, you'll have disruption of the growth of the brain. So at this point, you'll appreciate you won't have the gyri and the sulci of the brain. At this point, you'll have a smooth brain and you'll have an image on it. So this normal growth of the brain has been disrupted. But schizencephaly, you'll appreciate there is a failure of corpus callosum. So corpus callosum will be present. And due to that, now you'll have like a fused brain at this point. The cerebral hemisphere would be present at this point. Then for the holoprosencephaly, you'll appreciate this absence of the midline cleavage. So structures will be present, will be merged. So that means you won't have the symmetry between this, the brain vesicles. You won't have formation of some of the structures in the brain. For anencephaly, you'll have failure of development of the brain completely. So this might be due to genetic factors, maybe some of the teratogens and other things. Yeah, and then macrocephaly at this point, macrocephaly is now, macro means large or big, then cephaly means head. So that means oversize or enlargement of the head. So remember, hydrocephalus can be a type of macrocephaly because hydrocephalus is enlarged ventricles leading to enlargement of the head. Then encephalocele is actually the herniation of the brain tissue through openings in the frontal bone, orbital bones, nasal bones, and occipital bones. So this is a herniation that we'll see in a few minutes. So we can appreciate this one. So this one is hydrocephaly. Hydrocephalus can appreciate enlargement of the brain, can even appreciate the sunset eyes, and even this. Actually, at this point, they are pronounced. This is smooth brain, this encephaly. Can appreciate the way it's smooth. The normal gyri and sulci are present here, but are absent in this one. So this one is encephaly. We can appreciate in this we have schizencephaly. So we can appreciate this point, the ventricles at this point, they haven't developed. This is schizencephaly. Hydrocephalus, once again, sunset eyes. Can appreciate the way you have macrocephaly at this point due to increased intracranial pressure. And then at this point, the fontanelles and even the features haven't closed, and that's the reason why you have enlargement of the skull. Smooth brain on this other point, you can appreciate the cerebellum is formed well, but the brain is smooth. So this encephaly. And then on this other end, you have schizencephaly. It is articulate. So for holoprosencephaly, that means the midline structures formation is wanting. So this is a normal brain. You can appreciate it. Have on either side, you have the cerebral hemisphere are present, the lateral ventricles are present in this one. Everything is formed. So for lobar one, you'll appreciate that we'll have some of the lobes of the ventricles. They are somehow minimal, they are smaller. As you can appreciate. So for example, here, the lateral ventricles are somehow smaller. In semilobar, they're somehow minimized. You can appreciate they're even small. And then a lobar, that means the ventricles are actually very small. And that means now even the corpus callosum. So the corpus callosum at this point is this one. So it means it's present in a normal brain. So it's somehow lobar, that means it's present, but it's now, can appreciate here, it's formed on one end, but on this other end, it's not there. Then in this one now semilobar, that means it's present on one end, but on that one end, it's also diminished. Then on a lobar, like it means it's absent completely. So this hollow prosencephaly. So this hollow prosencephaly can even mediate with like the person who has um, two eyes, but not those two eyes are opening on one orbital, uh, on orbital part. So try to differentiate between holoprosencephaly and encyclopia. And actually, you can appreciate at this point, there is absence of the corpus callosum at this point. So this is it. And then at this point, you can appreciate this holoprosencephaly now. At this point, we'll appreciate the corpus callosum will be facing the brain, and then the eyes will open on one orbital structure. So as we finalize, we can appreciate now this is an anencephaly. This is an anencephaly, a failure of closure of the cranial neuropole. At this point, you can appreciate this macrocephaly. Macrocephaly coming and going due to hydrocephalus sunset eyes. You can see them there. And then this is now is the frontal encephalocele. So brain herniation. You see the opening of the frontal bone. So most likely the metopic suture. And then you have occipital encephalocele. So brain herniation on the outside bits and through the occipital bone. Then we'll have an encephaly on this other end. Is an encephaly. So a person with an encephaly will also have a calvaria due to failure of development of the skull. Then, as it may be finalized, you can talk about nasal encephalocele. So nasal encephalocele will be coming from the region, the nasal region. So you'll appreciate some of them will have orbital encephalocele coming from the eye, others frontal, others occipital, yeah, and such other things. So those are some of the sample questions, sample essay questions you can have in an exam setting. So with a need of a diagram, describe the process of primary mutilation, with the differences between primary and secondary mutilation, describe the derivatives of secondary brain vesicles. Then another one is describe the various brain fractures and their time of development. The third one is to differentiate between communicating and uncommunicating hydrocephalus, then to describe the various anomalies of the brain and their content and their embryological basis. Yeah, so I guess with that, just to wish you all the best in your exams. Can you like share and subscribe at your own point, comment, and ask any questions that people having. But point to appreciate is that just know that he who watches you doesn't sleep, he doesn't slumber, will always be there for you. So with that, I wish you all the best in your endeavors. Thank you.